Well, thank you so much. This thanks to Dave and John and Dan and Krish and everyone, Eric. This is really a wonderful opportunity to bring us all together. And I've prepared a presentation a little more on the lighthearted side, fun side as requested, with a little science smuggled in as well. And of course, this beautiful photo from Asilomar ENC several years ago, maybe early April, when the flowers were in full bloom. I love seeing the ocean behind you, Dave. And I'll talk about, in particular, about some bacterial work and how Solids NMR is uniquely enabling and addressing some of the challenges that's posed by understanding bacteria that you can't do with solution NMR or crystallography. So really the Solids NMR being uniquely enabling. And just to bring you to talking about bacteria in contrast to all the flowers and the trees and the vegetation on Earth, which by mass contributes about 450 gigatons of carbon. So they're the winners on Earth. After plants come bacteria, 70 gigatons of carbon. So this is this really awesome chart of all life on Earth by carbon mass. And if you're having a hard time finding where we fit into this picture, well, you've got to look really small. So all these cubes and squares are scaled to the amount of carbon mass, and humans are down here 0.06 gigatons of carbon, so really tiny compared to bacteria, 70 gigatons, second only to plants. And in contrast to the way people have studied bacteria in the lab for many years, just growing them in flasks or little culture tubes, bacteria really inhabit the earth and in the body and in different native niches as biofilms, as communities of cells. They're social creatures, just as we are, forming neighborhoods and cities where they form these little cities surrounded by these complex extracellular matrix materials that protect them and confer protection to this small city where they can divide labor and activities throughout the community. And you'll find biofilms everywhere if you look. So these acidic mats in Yellowstone, those colors come from the different metals and molecules produced by bacteria in these biofilm mats. You also find them in the body, sometimes a nuisance as dental plaque. Those are bacterial biofilms. They also cause a nuisance in industry, clogging pipes. And you also find them very beneficial. They're beneficial biofilms. For example, these rhizobia that form small nodules associating with the roots of plants, where they fix nitrogen from the air to make nitrogen-containing molecules that the plants use directly for their nutrients. I learned a lot about rhizobia. I went back when I was a graduate student, actually doing whole leaf NMR, growing soybean plants. These are soybeans, and to inhibit the production of these rhizobia, actually we fertilized the plants with N15 ammonium nitrate and then did solid-state NMR on those. Also flowed over the leaf C13 CO2, a gas mixture to get carbon-13 into the leaf. So where there's a will, there's a way with solid-state NMR and complex biosystems. And I'm going to tell you about our work on bacterial biofilms. We couple NMR with a lot of biochemical techniques and microscopy, which always makes for pretty journal covers. And you can tell those were more biological journals, but it was in molecular physics. We were able to smuggle in an actual NMR spectrum onto the cover. We were glad about. And so in our lab, we've been trying to push the envelope on using solids NMR, not really for individual protein samples, but these complex collections of materials, pushing the envelope on whole cell NMR, extracting specific information about the cell wall or even ribosomes. Oh, here's a picture of our lab. I'll show a few more photos to kind of, in this lighthearted way, introduce you to our lab. We hope we would see you in person here someday. Here's one example of how we do kind of macromolecular solids NMR. When we isolated intact ribosomes from E. coli, you can collect that NMR spectrum here. This is N15 labeled. And if you compare that to E. coli whole cells, you can see the whopping contribution of nucleic acids to a cell is ribosomal RNA. And so you can scale by those peaks and you get the right composition, the right percentage of the cell that's ribosomes. And so you could use these kind of signatures to track cellular metabolism over time. So we often do that type of approach. I'll be telling you about beyond the cell surface, our work on the extracellular matrix. And that's where we've been using NMR to look at biofilm composition, and also in our biochemical work to develop new anti-infectives to block the bacteria from forming these biofilms that are a real nuisance in infections. But the one example I'll talk about is where we actually discovered new chemistry in this complex matrix shown behind there in that biofilm mesh. Actually, we identified a new chemical structure lurking under those matrices that was never before identified in nature or even proposed as a possible polymer structure. So that will be the scientific example I'll share with you later. And here's just a picture, some pictures of our laboratory where we have two moderate field, or maybe now that's called low field NMR, 500 megahertz wide bore solid state NMR spectrometers. And we use our home-built all transmission line probes, the Jake Schaefer, Bob McKay style probes, where I learned from them how to use these and build them. And this was one group of students where they learn now how to, modify or sometimes repair parts of the probes. And here's some of those pictures in action. You could tell this is all pre-COVID, no masks, everyone's smiling. And this was up here at the top of the probe. Of course, that's the part that goes up into the magnet. And this is a former student who Dave spent a lot of time with, Joe Romaniuk, who just a couple of years ago started a position down at Dow, bringing the first solid state NMR spectrometer to their site down in Texas, and also doing solution NMR. And then this is more recent. I know the NMR field, we like to share stories, trials, and tribulations of not only working with the probes and the oscilloscopes, but also fixing equipment. This was, so you can tell now this is in the era of COVID, so all the masks, but still smiling, that's the important part. And this was repairing a power supply issue, which actually Dave Rice helped us with 5 years ago, this same issue revisited. So again, where there's a will, there's a way. You find out how to replace these Molex connectors that stick out there. You buy this crimping tool, and this is always a good sign. All the green lights are back on, and we're back in operation last summer. And this goes back, I thought I'd shout out to Dave Rice, who actually was in our group for about 3 years. What a wonderful time back several years ago now, time flies. And so you might sometimes feel like you want to pull your hair out, but still smiling, right? And that was our group then, that was Joe there. And just to show you, by way of introduction, the rest of our lab is a typical kind of bacteriology lab, biochemistry lab, where we do a lot of microscopy and biochemical experiments to couple with our solid state NMR discoveries. And so I'm going to just tell you a short story on one of our recent discoveries that I get really excited about, which is how bacteria called E. coli, which are found in the bladder and the kidneys, and one of the major causes of infections in the US and worldwide, one of the most prevalent infections. So with the largest patient population, just form these seemingly simple biofilm communities, but it's such a complex mixture. It's been difficult to decipher and determine exactly what makes up this matrix, and therefore how can we interrupt it and inhibit it. And to inspire the production of new materials, how do they make these robust structures? What polymers or molecular recognition rules, design principles are at play? And so I'll tell you about the types of biofilms of E. coli we've been studying that are really fascinating. They produce two major components, so they're not as complex as we thought, where there could be maybe 8 components. They're really two major components, polymers. And one is an amyloid fiber that bacteria produce at their cell surface. So in contrast with amyloids associated with neurodegenerative disease and Alzheimer's and Parkinson's, where protein folding goes awry, and you don't want the undesired formation of amyloids, bacteria actually harness dedicated machinery, genetic machinery and molecular machinery, to elaborate amyloid fibers at the cell surface for function, to adhere to one another. And this is the picture of that. They get very long, almost the length of the size of one of these E. coli cells. Together with a cellulosic component, then they're able to really enrapture the whole cellular community together with these protective nets that really protect them from antibiotics and stresses, desiccation, and it requires both of those components. And I'll tell you, I should have an asterisk up here on cellulose. I'm going to tell you about how it's not just regular cellulose, but a new kind of cellulose that we discovered. And so you all know cellulose is the most abundant biopolymer on Earth. You're probably all wearing some form of cotton today that's made of cellulose. Here's cotton, a cotton plant there. And because it's present in all the trees and the leaves on Earth, it makes it the most abundant biopolymer on Earth. And we make our paper and some sweets from it. And actually, the bacterial cellulose can be produced at high quantities, just like the tree type unmodified regular glucose polymers. And it's actually being used in a wide variety of products for its material properties, has slightly different physical characteristics than plant-based cellulose, even used as lightweight materials for electronics and for wound dressings, for example. And this is our E. coli again, where when you look at the micrograph, you cannot tell which part is coli, which part is cellulose, how much of each component is there. And this is where we sought to use solid-state NMR. But first we pulled out and extracted this slimy glue-like biofilm matrix material away from the cells to simplify the system and remove the cells. And so this is a simple schematic just to illustrate how we do that. After growing up lots of bacteria, we homogenize them with this kind of omni mixer that's like a kind of smoothie maker, just an immersion blender that through sheer forces strips away the outer material from the cells. And then we centrifuge, clean that up a little bit, and we have our pure material. And the first thing we did before NMR was just to look at this under the microscope. And we were really stunned to find that we could remove these kind of intact supramolecular baskets from the E. coli. So on the top left is one E. coli having its kind of sleeping bag removed from it, whereas we mostly remove the baskets away from the E. coli. And here down at the bottom is a collection of 4 of these. You only have to perturb the integrity enough so that the bacterium pops out, and we have these hollow 4 baskets. If there's still a cell in there, it's very dark, so you can tell when there's a cell or not. And this is on the order of the size of the 1 E. coli, about 1 micron. Again, you can't tell by looking at this how much is coli, how much is cellulose. So this was where we took a solid-state NMR approach. And this is just simple C13 CPMAS NMR. I'll give a quick primer on that in a second. Natural abundance levels of carbon, about 40, 50 milligrams of material probably was in our 5 millimeter rotor. And here's the carbon spectrum of that intact matrix material collected. And we also prepared separately the cellulosic material that the bacteria make and the proteinaceous amyloid component called coli that they make. And we took a simple approach and took the spectral sum of those 2 polymers, scaled, and found it was the perfect recapitulation of the extracellular matrix, really proving to us it was only these 2 major components that make up that protective basket. And we were able to quantify that it was mostly coli, 85%, but required this 15% of the cellulosic material. And of course, at the time when we published that, we also made a physical mixture, with 85 milligrams of coli, 15 milligrams of cellulose, and you get a triply degenerate, same spectrum overlap there. And what drew our attention at the time and took the better part of a couple of years to really nail down was the identity of this kind of mystery peak that always made us very curious. And it was a good thing we were using our bacterial cellulose material to identify what that was, and not just commercially available plant-based cellulose. And before I show you how we determined what that identity was, let me give the quick primer, because I know so many on the call on the Zoom are more of the solution NMR aficionados and small molecule NMR. So just briefly, how is it that we're able to obtain these spectra of these intact and tractable baskets? As you know, if you try to obtain a solution NMR, a spectrum of it by solution NMR, your lines would be so broadened as to be indistinguishable from the baseline. You wouldn't see any spectra, and that arises from the contributions of dipolar couplings and chemical shift anisotropy, whereas solution NMR requires your molecules to be tumbling rapidly isotropically. And so in solid-state NMR, we mechanically spin our samples to achieve this type of averaging. And then we have these spectra that we can interpret. And of course, we're spinning at a precise angle called the magic angle, which corresponds to the body diagonal of a cube. When you rotate a cube about the body diagonal, you rotate the Z into the X into the Y axis. So you can appreciate you get this spatial averaging. And this is really combining cross-polarization with magic angle spinning as one of the major platforms for most biosolids NMR experiments introduced back in 1976 by Schaefer and Stachkal, where you can see with just cross-polarization and some proton decoupling, it's difficult to interpret the line shapes, but by implementing magic angle spinning, you start to rival that of a solution NMR spectrum, except you can study the intrinsically insoluble solids that cannot be studied by solution NMR. And there's something also aesthetically appealing about the magic angle. Artists think so too. This was a beautiful cube hanging from its magic angle. I saw at the Ontario Art Gallery a few years ago when visiting the students, the biophysics students at the University of Toronto. So I hope you'll always look at the magic angle and cubes in new light. We also have in the lobby of my building here in the Keck building at Stanford, a Pomodoro bronze cube perfectly positioned at its magic angle. And I just wish keep petitioning my chairman if they could put this on a rotating platform so we'd have the Pomodoro spinning at the magic angle. So now I'll come back to our science problem here. And that was, what was the identity of this mystery peak? In regular cellulose, it's a polymer of glucose 6 carbons that has a very representative spectrum. Probably some of you are familiar with this one in particular of cellulose or glucose polymers. And ours always had a 40 ppm peak consistent with an amino ethyl type carbon and amino type carbon. And Weary, a graduate student at the time, really cracked this case open, and we discovered that the cellulose was modified with phosphoethanolamine. And we did a number of experiments to prove that. One really terrific, exquisitely tuned one is to use the Redor experiment where we measure dipolar couplings. And I didn't mention this back here, but for the aficionados, this is the very short truncated versions of the Hamiltonians for chemical shift anisotropy and dipolar couplings. And the dipolar couplings contain crucial spatial coordinates and spin coordinates. And although we're averaging over the spatial coordinates through spinning, we can manipulate those interactions by manipulating the spin coordinates with pulse sequences. And we use Redor to defeat that averaging and reintroduce the dipolar coupling. And there are, hundreds of different ways of sequences of how to reintroduce dipolar couplings. And we used Redor here. So you could see, for example, the C8 carbon we label up here would be one bond away from the nitrogen, have the strongest dipolar coupling. And you see that in our Redor difference spectrum. And through a number of experiments like that, also to phosphorus, we determined that it was phosphoethanolamine cellulose, and about 1/2 of the glucose units are modified. Also something that requires solid-state NMR to determine. And if one tries to digest this material and look by mass spec, you lose all the modification, cleaves off, it's labile, and so you can't use harsh digestion approaches in order to determine this structure. And so this also explained why our cellulose had this hydrogel-like material that would stay in this thick gel-like solution, whereas regular cellulose would precipitate out. And so this begged the question, how on earth does E. coli install this modification? This was the first identification ever in nature that an organism can make a modified cellulose. And again, cellulose is the most abundant biopolymer on earth. So it was such a surprise that this had just gone unnoticed. And for decades, people had been studying these E. coli biofilms, and it escaped detection because of the conventional methods that were used that lost that modification. And so this presented the first identification that there's a modified cellulose. So of course we had to answer, how do E. coli install it? What gene or genes is involved in making that? And so we took an approach and just made some mutations in genes that were associated with cellulose synthesis, but were kind of mysterious. There was a real craze in trying to understand what are the roles of some of these genes that had no known function. in contrast to the main machinery, AB machinery, that's known to make the polymer. And we found that the BCSG gene or enzyme then was specifically required. And without that, you lost the modification and you resorted back to just regular unmodified cellulose. And we later went on and did more biochemical experiments and proved that this was a phosphoethanolamine transferase, an enzyme that also before had no known ascribed function. And it's required for biofilm properties. So when bacteria are forming biofilms, you can grow them on an agar plate and they elaborate these hallmark wrinkles like this. This is, the size of a small coin. You could see this by eye if you were looking at it. And without the modification, they don't wrinkle. So clearly this zwitterionic modification, phosphoethanolamine, is required for interactions with those coli polymers. And this was kind of a cartoon of how this machinery comes together. A and B, there were structures for at the time. That's the main cellulose synthesis machinery, getting cellulose produced out into the periplasm. And this kind of schematized cartoon, this beautiful one was in a perspective article that accompanied our article. We had a much more simpler kind of block diagram, but this was very nice virtual pictures of what those proteins might look like. And with a title, I wished I had thought of a bacterial coat that is not pure cotton, because not just regular cellulose. And that showed our enzyme, BCSG, they're tacking on this red modification. And now in later work with Joachim Zimmer at UVA, well-known crystallographer, now cryo-EM guru, we were able to help him on his quest to do cryo-EM of this whole entire complex. And he further refined the understanding of that cellulose machinery, synthesis machinery, that one partner, BCSB, forms a kind of hexameric assembly. And here's our BCSG. He sees it as a dimer in this structure. So that might allow a modification from each phase or each side of the cellulose polymer. And of course, moving onward, we're really interested on the solids NMR side to understand the kind of design principles and how bacteria take these two different types of insoluble fibers, modified cellulose fibers and coli, and elaborate these connective, cohesive, kind of elastic structures made of the coli and the cellulose put together in the way a basket weaver would weave a basket together. And we're doing this through a variety of spin diffusion methods. Also, Redor can help stitch together the contacts between these and these really heterogeneous non-crystalline assemblies. And there's also, as I mentioned, these applications for the unmodified bacterial cellulose that are known. Now we have this new chemically modified cellulose that might have very many different types of properties, materials properties that we hope people will consider and studying. And we're trying to think of our own applications as well. What would be the best ones to potentially use this for? So I'd like to thank again, the organizers for this opportunity, for everyone for joining my group here. And these were the specific people who contributed to the biofilm NMR work. And of course, our Dave Rice here. So thank you.
Impressive lineup of transmission line probes.
Thanks.
Jake Schaefer written all over it.
Yes, we have HFCN and the HPCN. I'd be happy if there are any questions or.
Any questions?
Lynette, can I ask you a question? This is Manda from India, from Hyderabad.
Hi, yes.
Hi. So the point here is that the plants have eventually evolved to break down the biofilms, right? The modified cellulose, isn't it? Otherwise, the evolution of plants, particularly the plant roots, would have been in deep trouble. So how did plant combat this? Do they have a special enzyme, set of enzymes that would break down these biofilm forming celluloses?
Yeah, well, plants don't make the modified cellulose, it's just by bacteria.
I understand that. That's what I am asking you, that plant has to combat the biofilm that has been formed by the bacteria, right?
The plants don't, yeah, the plants don't combat these biofilms. So these biofilms from E. coli and Salmonella can actually infect plants. So when you have this alfalfa and spinach outbreaks, caused by E. coli 0157H7, they are making these type of biofilms. And so plants don't break them down, but we have a whole study on a kind of bioengineering side of cellulase enzymes produced by fungi that can degrade the bacterial cellulose. But it's not, they don't do a great job of our modified cellulose, more for the unmodified glucose units. And we're looking at that as an inhibition strategy for blocking.
So it means that whenever this pathogenic bacteria evades along with the biofilm to the plants, there's hardly any way the plant can survive through that, right?
Right.
So the plants haven't evolved so far to combat the biofilms. That's what you want to say.
Not these with this cellulose and coli. So those ones that you've read about, the outbreaks like the one in Europe several years ago caused the demise of lots of farms and farmers. Those were really devastating because they were making these virulence factors, especially those coli amyloid fibers, whereas we think the cellulose tends to mask the effects of that coli. So there's a balance, and we have a recent paper on that, to how bacteria tune the balance of the amyloids and the cellulose for different functions.
Well, thank you so much. Wonderful.
Sure. Thanks for your question.
Can I ask a question?
Yes.
So that was really great talk and super interesting. So when you've deleted this gene and it can't put that modification is, do you see a different appearance in the microscope of the cellulose? And have you figured out how the properties, a little bit about how the properties differ?
Yes. Yeah. Let me show. I think I have just one picture here that speaks 1,000 words.
This one's just hitting 100,000.
Oh, you can't see it anymore. Okay, I'll just share really quickly there. Yeah, here you can see with the regular, without the modification, that's the mutant down below. And this is just a green stain that lets you see the cellulosic material. They're really short and stubby fibers, but when it's modified, you get these elongated fibers, like they're able to entangle more with that modification, maybe even, you know, making them appear longer. So you can see the difference at the really close level. It's interesting. So
That's really cool. Thank you.
thanks for the question.